decided in the program to include a couple tutorials. Simon will give one on modeling, and I'm giving this one on electrophysiology recordings, specifically about spike sorting. If you know what spike sorting is and you want to take a break, I will be talking about spike sorting and why it's important. Yeah, I see you guys can go. So, if you don't know what I'm talking about, if you work in cognitive neuroscience and you don't do recordings, the idea is to try to give you something that you'll have a clue what goes on in the type of data you will see later on in different talks. So if somebody's showing data from rodents or from monkeys, or as I will show from humans in the afternoon, you know all the stuff that goes into showing how these neurons fire, because it's not like, like plug and play and you get the results. If you are doing spike sorting and you don't know what you're doing, well, I will try to explain you a little bit what are the steps involved in processing the data. So, I have to cut a lot. I'm planning 1 hour thing. Let's start from the very beginning. So, it's a bit boring, but it's something that it's a very interesting thing. I study physics and math, so I come up with these things. It's very boring. I like it because I'm a physicist. But if you're a neuroscientist, it's very boring. But if you're doing it right, suddenly it's like, whoa, it was worth the effort. And I will try to show you this. At least that's my experience, and I think it might be the experience of many people doing it. If you manage to do a good job, suddenly you will start seeing things that nobody else saw before. That's a nice catch. That's a nice trade-off of this. So, but I try not to get too technical. But the very basic thing, let's start with the very basics, is if you want to record from neurons, you have to put an electrode inside the brain, right? So that's what we do in animals. We just drill a hole and we put an electrode, and then we record from neurons. And you just lower an electrode into a particular area that you are interested in with a particular animal you are studying. The electrode may have a tip. This diameter might be like a few microns, so it's a tiny fraction of a millimeter. And you get close to a neuron, and you will hear the neuron's activity. So basically, if you record the data with some amplifier, some equipment you buy, you may see something like this. And then you see some background activity, which is the noise. And the noise is not physics noise or thermal noise and so on. The noise is mainly composed of activity of neurons that are far away, right? So you're hearing them. But the ones that you see clearly are these things here, which are the spikes. And these are the action potential. This is the firing of the neurons that are close to the tip of your electrode. By the way, the electrode is completely isolated except on the tip, right? So you record only from here. You don't record from around here. All right. Now let's see this process in more detail. So let's do a cartoon. So if I put an electrode and I'm recording from a set of neurons, what happens? I amplify the data, and that's exactly what happens when you have your whatever acquisition system you have an amplifier, you're recording against the reference, and you may see something like that. And that's actually real data. That's how real data looks like. That's what you get from your electrode amplifying the signal. Now, there are two things you can do. The first thing is you can low-pass filter the data. So this is broadband, no? It's a signal that has activity from 0 to thousands of hertz, right? If you low-pass filter the signal, so you stay with the low frequency, typically between 0 and a few hundreds, that's what people study. This is what we call the local field potential. If you are old-fashioned, that's what we call intracranial EEG. It's local because it's recorded by a tiny electrode, so you will get a signal here and not from a huge electrode that records more average activity. And typically we analyze this signal. We see like the frequency composition, the power spectrum, if we have activity, if we have some rhythmicity in the data, and so on. This is very well studied, for example, in rodents. If you study hippocampus, you will see that there are some oscillations in what is called theta band, around 8 hertz, 10 hertz, and we know a lot about this. Now, if you get the same data, and if you high-pass filter it, then you see that. You see the spikes on top of the background activity, and the spikes are the firing of these neurons. Actually, these spikes, if you have a good eye, you will also see them here. They're very, very tiny, but you cannot detect them at all. So some of them, you will see in the recording. So in order to see this better, you high-pass filter the data. And now you can see the firing of the neurons. Now the problem is that when you record and you see the firing of the neurons, you see the firing of all the neurons together. And you don't want that. It's like if I put a microphone here and I start recording and you are all talking at the same time and I'm just hearing all the voices together. If I want to understand anything of what's going on in this room and if you're all talking, I should try to separate who is who from the voices. I mean, the voice of Simon is different to the voice of Silvia, is different to your voice and to your voice. So I will try to come up with some mathematical way of splitting the different voices to try to understand what's going on. That's called actually the cocktail party problem. It's a very well-known problem in mathematics. I mean, like we try to split the voices from one or several microphones. It's a similar problem here. If I record from this microphone, this electrode, and I'm listening the firing to all these neurons, well, I want to know which is which. It is a spike from this one, it is a spike from that one, and so on. Now, what saves us, up to some extent, is the fact that different neurons, they will fire spikes with different shapes, right? Now, if I get one of these ones and I expand it, you will see, this is in data point, this is about 2 milliseconds of recordings, this thing is 5 seconds. You will see that these spikes, they have different stereotypical shapes. Why is that? Because neurons tend to fire spikes of different shapes. I will explain you this in a second, why is that, right? So if you record there, one neuron will have one shape, another neuron will have another shape, and so on. This doesn't always happen, but this is our assumption. I mean, we start from that. If this won't happen, we're lost. There's nothing we can do. Now, this red, green, and blue, and cyan correspond to these three neurons, the ones that are close to our electrode. But neurons that are further away, the signal is a bit lower because they're too far, right? So the amplitude is smaller, and they are more contaminated by noise. In technical terms, the signal-to-noise ratio is lower. We don't have much signal. So these neurons here, in this annulus here, we cannot split anymore. We cannot really tear apart the shape of one from the other. And we group them together and we call multi-unit activity. We cannot separate it. And the neurons that are further away, we don't even detect anymore because they are too far. So these are the neurons that may have spikes that are below my detection threshold, which is this red line. For example, these tiny things here or the noise. So the neurons that are further away, they contribute to the background noise that I see in my recording, right? So that's the job we want to do. We want to split the different shapes. Yes?
Do we know the number of
neurons? No. That's the problem. Because if I give you this problem, and you are blind, you don't know how many neurons you have, it's much, much harder. Because you're blind. I mean, like, you don't know if you should get 3 different shapes or 20. And I will talk about this at the very end.
How can you define that circle?
No, that's a cartoon. Well, actually, I can define the circle. I mean, I didn't do that, but basically what you can do is, Pushaki did that, I mean, and other people. So you come very close to the neuron. You nearly touch the neuron. You patch it, right? Then you get a very high spike from this neuron. And then you start to retract the electrode. And then you see how far you can retract, and you're still recording from this neuron, and you can still separate it from others. This is about 60 microns. And then you keep on retracting, and when you keep on retracting, you are seeing the spikes of these neurons, but you cannot split them for the other neurons. So that goes from 60 microns up to 100 microns, or so 120. And if you keep moving forward, backwards, then you don't see the neuron anymore. Okay, why do spikes fire different shapes? Well, because spikes are different, because neurons are different from each other. I mean, they have slightly different physiological properties, and also because they have different dendritic trees. You know, the neuron has an axon and a dendrite, and the dendrite, there are branches of dendrites, and all these branches of dendrites contribute to the potential you record. Right? So if I put my electrode here, for example, I will record a spike like this, from this neuron. But if I put my electrode there, I will record it like this. So depending on where I'm recording from, with respect to the neuron, the spike of this neuron will look different. And the trick is that we have different neurons around, and I put an electrode, and the neuron will fire a different spike shape, which depends on where I am, but it will be different for the different neurons. Right? That's more or less what saves us, the fact that we have different spike shapes from different neurons. Yes? Happy birthday, by the way. I told you. So now we all remember his question. If I'm interested in local fields, I will go say, depends on the frequency you're interested in. I mean, I will not show you data, but we are interested, for example, in high gamma activity, which is, in our case, goes up to 150 hertz or so, from 80 to 150. So the low-pass filter has to be at least 300, because of, otherwise you start introducing distortions, aliasing, and so on. If you're interested in even higher frequencies, like fast ripples that have been described to be 200, 300 hertz, well, you better, start higher. So your low-pass filter will be maybe 1K, 1,000 hertz, from 0 to 1,000 hertz. Depends on your interest. Now, if you're interested in spikes, you can start high-pass filtering from 300, and you're fine. You kill most of the EEG activity, and now you can see the spikes very clearly. Okay, now, how do we do this separation? There are many, many methods. That's the most trivial one, but I just show you this because that will give you an idea of how things can be done. This comes, if you know acquisition system, I think this still is implemented in Plexon, in the type of system. So basically, here you see by eye that you have two different spike shapes, right? But what you can do is you, by hand, draw some rectangles, and you say whatever spike passes this rectangle and this one, it will be a certain spike and a certain neuron, and whatever passes this one and this one will be a different neuron. This way, by hand, you can separate the spikes of the different neurons. This is something that people still uses a lot, actually. Now, this is fine if you have one electrode, right? 2, 3, 4, maybe up to 10. But if you have like 100 electrodes, you cannot do this job. And the problem is the recording change with time. They do change with time. So the windows, you need to actually move them. You have to recalibrate them because the spike shape, they might change slightly even within a half an hour recording, right? So you cannot possibly do this if you have too many channels. The second problem is that this is a cartoon example. The spike shapes will never look like that, so clean and so separated. Typically, will be overlapping with each other. It won't be that easy to separate the spike shapes. So how exactly you draw your windows? So you have to make a decision by eye, and that's very subjective and very prone to errors, right? This is the problem with many of the spike sorters that we have had until 10 years or so. Another way to do it is you just say, well, you get the spike shapes, you get some features of the spike shapes. For example, you take principal components and you say, well, let's take the first two principal components and let's plot it against each other, and then you make it clusters. So these spikes are all the same, these spikes are all the same, and so on. And then by hand, you may draw an ellipsoid separating the different clusters. But it's the same problem. You have to do this by hand, right? And sometimes these clusters may be quite overlapping, so you don't know exactly how to draw the ellipsoid to separate one cluster from the other. So that's called from a more automatic solution. You don't want to be doing things by hand because it's very subjective, prone to error, and you cannot do it if you have many channels. And I will tell you the steps of how we do it, at least semi-automatically. Now we have a fully automatic algorithm, but I won't talk about this because it's more tricky. And I will particularly tell you about the algorithm we use because it's the one I can describe the most. But the steps for any spike sorter, you will see they're more or less the same. So it's more or less trying to solve the same problems. Okay, so the one we use is called wave class because it uses something called wavelets. I will tell you what wavelets is in a second. And it's freely available. You can download it, you can play it. And we have a web page where you can put questions, and so on. We have pilot data. You can use it with our pilot data. We have tutorials. But I will explain you how it works. So before that, there's a big problem, and that's related to your question. How can we test a spike sorting algorithm, right? Imagine that I come with my best ideas, and I put them together, and I come with my spike sorter, and I say, this is much better than anything else. Well, how do I know that? If I get a real recording, Simon has his spike sorter, and he said, well, from this recording I can get 3 neurons. And then I said, well, actually from the same recording I use mine, which is much better, and I can get 20. And Simon will say, well, how do you know you're right? Maybe you are just doing it wrong. Maybe you are just splitting things that you shouldn't have split in 20 clusters. And since it's real data, we don't know. We don't have ground truths because we do not know from how many neurons we're recording from. right? It's real data. I don't know if I have 20 neurons around it or if I have only 2, right? So one way to test these algorithms is to do simulations and to simulate data, real data, as best as we can. So I will tell you what we did. I mean, it gets very tricky also, but I will give you like the first level of simulations, which will give you the idea. So basically what we can do is we can generate background noise. How do we generate background noise? You don't just use Gaussian noise because the data is not Gaussian, right? So the way we did it here is we got spikes of different amplitudes and of different shapes, and we superimpose them to each other millions of times. And when you start superimposing spike shapes, so spikes like these ones here, when you start superimposing to each other millions of times, then you start getting something that looks like noise. Actually looks like the noise you get from the real recording. You can describe the characteristics of this noise, the noise from real recordings, and it's very similar. Power spectrum, amplitude spectrum, and so on. And on top of this noise, we put spikes that we know. So I know that I'm putting a spike of this shape in green, here, here, and here. And I know that I'm putting this red one here, here, and here, and so on. So if I know exactly which spike I'm putting and where, then I have a ground truth to quantify how good or bad my algorithm is. And then we can play with this. We can do simulations in many different ways. We can put higher levels of noise. We can use different spike shapes. I mean, this is a very simple example. This is a harder one because these spike shapes are very similar to each other. And we can test our algorithms. So keep this in mind. I will come back to this at the end. Now the code. How does it work? How do we do this idea of spike sorting? Well, there are 4 steps. I call the first step step 0, which is a step that I used to ignore before, and I realized everybody was ignoring, but which is quite interesting. So the first thing is what I told you. Yes?
So can I just ask a very high-level question to make sure I'm following this?
Yes.
So the real situation here is that you insert an electron and you record data, but you don't actually know how many neurons you have.
Yes.
So the goal of the whole thing is basically just to determine how many neurons are you actually recording from and which aspect of your signal can be attributed to each of those neurons. Yes?
Which aspect of your signal noise? I'm recording spikes, and I want to know first how many neurons are contributing to my recording, and then which spike corresponds to which neuron. And that's called spike sorting. I'm sorting them. I say, okay, this is neuron 1, this is neuron 2, this is neuron 3, and so on, right? Now, how do we do that? 4 steps. And let's go one by one. The first thing you have to do is you have to filter the data. This is what I showed you before. Like several years ago, I didn't even mention this, but now I mention it because there's something quite interesting with this process. If you filter the data, well, when I started doing this, I was a postdoc at Caltech, and I came from math, and I was seeing the spikes in books, and I was seeing the spikes that everybody was getting in their acquisition systems, and they more or less looked like that. So you get the polarization, and then followed by an hyperpolarization. So you get an excitation and a rebound. So it goes back, and then it goes to baseline. And informally, many people were saying, well, that's actually how it looks in intracellular recordings. I mean, we know that you have a first depolarization and then hyperpolarization. And some people, not all, were actually commenting on this. I mean, actually the first derivative of the intracellular potential, if you get inside the neuron, right? So I was looking for spikes like this, and I started doing my first recordings. I mean, I came from math, so I started doing my first recording in humans, and I will tell you this in the afternoon, and nothing worked. I mean, for me, everything was wrong. I mean, I didn't understand what was happening because my spike shapes, they look like that. And I'm like, that's a funny triangle. Doesn't look like the spikes I see in books. I mean, like the spikes I see in books, they have this rebound here, which I never found. I never saw that. Actually, it happens, but it's very rare. Now, what it took me some time to realize is that all the spikes I was seeing in books, they're actually filtering artifacts. They're not like that. So, and that's a problem because people, to record a spike, they use something called causal filters. The idea is very simple. If you're recording, imagine you're recording like this. You go like that. So times 0, and then you're recording. And this is how the data looks like. A causal filter means that every single data point that you have, you filter based on the previous ones. A non-causal filter means that every single data point, if you're going to filter data, you filter with a window around the data point. Now, the problem we had, at least 10, 20 years ago, is that this takes a lot of storage because the recording has to be done with 30 kilohertz, 30,000 samples per second, which is a lot. And if you're recording from any channels, you start filling up hard drives very quickly. So what people did is they were filtering online using causal filters. Because if you're recording the data online and you just got this data point, you want to filter it straight, but you don't know what is coming next, so you have to filter based on the previous points. This forces you to use a causal filter, right? Why you want to do that? Because if you filter online, you just put a threshold, and the only thing you keep is the spike shape. If there's a spike, you just keep the spike, and you throw away the raw data, because it takes too much storage. Now, it is well known in signal processing that a causal filter tends to distort the shape of whatever you're filtering, right? And this is because, if you're more technical, this is because the causal filter has a nonlinear phase relationship. The phase is not linear of the filter. But doesn't matter. I will show you this with an example. So to convince myself, what I did is I built some simulations the way I was telling you before, and I put these spike shapes, the one that I was finding. I put them in my simulation, I detect them, filter them, cluster them, and then I was getting this. Okay, these are the spike shape I put in my simulation, fine. Now, if I was using a causal filter, as everybody else was using, then I started seeing like that. And if I was increasing the high pass filter up to 600 or 900 hertz, as many other people use, then I started seeing things as in the books, right? So the first thing is, well, filter is important because if you don't do it properly, the spike shape you get is highly distorted, right? And it was tricky also because sometimes we saw stuff like this. Does it look all right? Does it look like a spike? Just a second, let me show this. Actually, if you do the proper filter, and this is filtered with a causal filter, if you do a proper filter, the signal is like that. So this is clearly an artifact. But if you filter it not properly, it looks like a spike. Or it may look like a spike, yeah.
So when you talk that you use non-causal filter, does that mean that you use it offline?
Offline, yeah. There's a way to do it online, which I propose, but I mean, we have to convince the people building the system. You can have a little buffer. So technically, you won't be online, but you can be delayed. You can be lagging by, say, by 500 milliseconds or so. And then you can record data and filter it with a little delay. Okay, I will hurry up because I want to finish in 20 minutes. Now, the next step is, okay, with filtering is fine. We have to detect the spikes. And there's a little trick also. If I have the data, let me see if I have data here. Yeah, if I have this data, right? And if I had to put a threshold to detect the spikes, I will say spike is whatever crosses this threshold. What people used to do, if you want to do this automatically, many people will put it by hand. The people that were doing this automatically, they will calculate the standard deviation of the data, and they will say, well, my threshold is at 2.5 or 3 or whatever number of standard deviations of the data. That has a problem. The problem is that the standard deviation of the data also considers the spikes. So if the spikes are huge, because you are having a good day and you're getting a very nice recording, the standard deviation goes high, because the spikes are contributing to the standard deviation. And actually, if the neuron is firing a lot, if you have many spikes, the standard deviation goes even higher. And that's something we don't want, because I want my threshold for detecting spikes to be locked to the noise, irrespective of the spikes I have. And there's a trick in mathematics, and that's from wavelets. Well, it's used a lot in wavelets, that is a way to calculate the standard deviation from the median of the absolute value of the signal. So believe me, that's a trick. It's a theorem. I mean, you can prove it. But if you take the median of the absolute value of the signal and you divide it by this magic number, this is an estimation of the standard deviation of the data, if you have noise. Now, if I have spikes in my data, doesn't matter, because the spikes don't contribute much to the median. The median stays more or less the same. So the nice thing is that if I put a threshold, which is 4 times this estimator, instead of 4 times the standard deviation, I'm much more robust. And I show you this. I did it with simulations. So I had a simulation, so I start putting more and more and more spikes. So I start increasing the firing rate of the neuron, my simulated neuron. And this is how the estimation based on the standard deviation went. So it started with higher, 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 and the one using the median stayed very close to the theoretical value, which was 1. So it's much more robust. So it's a very simple trick. So we have 2 tricks so far. Use non-causal filters, use an estimation based on the median to put the threshold. All these tricks, I mean, like if you want to use our codes, they are already implemented, but I just want to tell you all the steps that are there. So I skip that, we don't have time. The second step, so we have step 0 filtering, step 1 detection, step 2 is feature extraction. We want to extract features of the spike shape. I don't want to put the whole spike shape into a clustering algorithm. What are features? Say, for example, I can take the amplitude of the spike, I can take the width of the spike, I can take principal components, I can take the energy, the power of the spike. I mean, there are many ways you can define features. I use wavelets. Wavelets is, I don't have time, I had a slide about wavelets, but I think I will try to explain it with my hands. Imagine a Fourier transform. A Fourier transform is describing a signal from the time domain, it goes into the frequency domain. If you have a sinusoid that is varying in time, you do the Fourier, and you describe this, well, I have a peak at 10 hertz, whatever. Now, wavelets is a time-frequency decomposition. So basically, it gives you frequency activity, but at different times. So now you have a 2-dimensional representation. In one axis, you will have frequency. Actually, if you want to be more technical, it's called scale, but doesn't matter. And in the other, you have time. So basically, what you need to know, I mean, there's a lot of theory. There are many books about wavelets, but the important thing is that if you have different spike shapes, the different features of the spike shapes at different times, for example, this little bump here will be reflected in different wavelet coefficients. I said in another ways, there will be a wavelet coefficient that will characterize a bump with this frequency content and at this time. There will be another wavelet coefficient that will characterize this decay, which is different here than here. So you have different sets of wavelet coefficients characterizing different features, localized features of your spike shapes, right? So, sorry, but I have to skip this. So basically, the idea is the following. You start with the spike shape. These are the different spikes. See, I have like 1,000 spikes that are like this, 1,000 that are like that, and 1,000 that are like that. So this is the spikes in the time domain. If I do the wavelet transform, I get a set of wavelet coefficients. You can do something called the dyadic wavelet transform, which is a non-redundant representation. All this means that if you have 64 time points, you get 64 wavelet coefficients. Non-redundant because you get exactly the same number of coefficients as time points you had before. And non-redundant also because you don't lose any information. All the information in the spike shape will be in the wavelet coefficients. So if you do that, it's just one line in MATLAB, you get that. You get the distribution of wavelet coefficients. So for example, these will be the low frequency coefficients, they will be the high frequency coefficients. And the interesting bit on this is the following. There are some coefficients of some particular frequency and at some particular time that you see that are distinguishing different clusters. So there are some that they are crappy because they have more or less the same value for all the spikes, but there are some that they're making some separation. There are some that have high value for this coefficient and low value. Well, these are the good coefficients because they are separating different type of spike shapes. Now the problem is I have to choose these good coefficients automatically, and that's the tricky bit. So how do I do that? Well, imagine that if you have a coefficient that is crappy, the coefficient is not really distinguishing different type of spike shapes. The coefficient we have, in principle, a unimodal distribution, which might be Gaussian. And if you have a coefficient that is separating different clusters, well, then you will have a multimodal distribution, as I showed you before. So how can you distinguish these two things automatically? Well, you go to math, and there's something very well-known called the Kolmogorov-Smirnov test for Gaussianity. You can test how Gaussian is this distribution. And if it is non-Gaussian, it's good. If it is Gaussian, it's bad. So you can run this test, and basically you keep the coefficients with a known Gaussian distribution. And by doing this, you are getting rid of many coefficients that they don't help you to separate spike shapes. So you are reducing the problem. That's called a dimensionality reduction. And that tends to help. Okay, and the third and final step is, okay, we detected the spikes, we extracted a few features of the spikes, so a few Webley coefficients, and now we have to cluster that. We have to say the spike with these values of Webley coefficient correspond to neuron 1 and this other to neuron 2. And for this, we use an algorithm that comes from statistical mechanics, was developed by somebody called Eitan Domani at the Weizmann Institute. And it's basically a nearest neighbor algorithm. And I will give you again the gist of the algorithm because it's extremely powerful. So let's do it like this. Let's skip the theory. This example is as hard as it gets for clustering. This is an example that is very challenging for any clustering algorithm. How many clusters you see? How many groups of points you see there? 3, no? But it's not that easy because you don't have one blob here, and one blob there. So if you have 3 blobs here, something like k-means, which you might hear about, it's very easy. You say, well, I have 3 clusters. You put one center here and some variance around, one center here, one center here. But here, the center of this cluster is here, which is outside. Actually, there are no points here. And the problem here is that the distance between members of the same cluster is larger than the distance across clusters. So that's a very hard algorithm. The trick of this algorithm that I say superparamagnetic clustering is that, in essence, it's a nearest neighbor algorithm. So we'll not try to find the center and do some variance around the center. Will be based on nearest neighbors interactions. So basically, we'll say, well, these 2 points are from the same cluster because they're nearest neighbors, but these 2 points are not because there's a big gap between these 2 points. So if there's a gap, they don't belong to the same cluster. If there's continuity, they belong to the same cluster. Well, why these 2 are from the same clusters? Well, because there are many points in between that connects them, right? So there are many nearest neighbors around that connects them to each other. This is just the gist of the algorithm. And if you try it with this recording, well, with this toy model, I mean, you can do it perfectly. K-means fails completely. I mean, like standard clustering algorithm, they will fail miserably with an example like that. OK, so to wrap up, and I want to show you why we do this, and then I will stop. 4 steps. So first, you have to filter the data. We have to use non-causal filters. Then we have to detect the spikes, and then we use this threshold, and we put something, an estimation based on the median of the absolute value of the signal instead of the standard deviation. That's worked very well. We extract features, and for this we use wavelets. I didn't show you this, but we show it works much better than PCA, than anything else that was around at the time, and I think that's still around. And then we do an automatic clustering based on this superparamagnetic clustering algorithm, and then we can split the 2 different spike shapes. And this works very well. And I want to show you a preview of the talks in the afternoon, because there's a technical side to all the memory stuff that we will see in the afternoon. The technical side is this, and I won't repeat it in the afternoon, so we'll get rid of this part. We do recordings in patients. So we do single-cell recordings in humans. These are patients that they are epileptic, and they are candidates to epilepsy surgery. This means that they are potentially going to remove the bit of the brain that starts the seizures, right? And in many cases, for doing that, they have to put intracranial electrodes to try to localize as best as possible where the seizures come from. They don't want to remove the wrong area. So sometimes they will put these intracranial electrodes, which they look like that. I mean, this is about 1 millimeter thick. And they want to record the seizure activity to identify which is the area that is starting the seizures. And depending on the function of this area, they may remove it or not, right? So these electrodes, the ones we use, they were developed in California by Itzhak Fried at UCLA. They have microwires at the end, and from this one we can record single cells. Now, I say it now, I will repeat it later. The reason why human subjects have electrodes inside the brain is clinical, because they will put the electrodes there, the surgeons, to try to cure them from epilepsy. But this gives us an amazing opportunity to record single neurons in humans, and that's what we do. So I show you a real data. So this is a recording from one of these microwires, and you see you have many spikes. This is real data from a human neuron or human set of neurons. You put the amplitude, you detect the spikes, then you do the spike sorting using this method that I show you, and you see that you have different spike shapes. Actually, we got 3 or 4 more. I mean, I just plotted you 5. This is multi-unit, and these are all single units, and there were 3 more that I'm not showing you. If I don't do spike sorting, if I get all the neurons together, all the spikes together, this is how it looks like. These are all the detected spikes. Now our experiments, I will show you more in the afternoon, is basically patients will see pictures, and then we see how the neuron fire to picture presentations. And for example, we show these 5 pictures. Actually, we show more, but I'm showing you only 5. And if you take the detected neurons, this is times 0 more or less here, you don't see anything. Doesn't seem to be any response here. Now, if I will show you only the spikes fired by this neuron here, cluster 3, look what happens. There's a huge response to Vladimir Putin. That was times 0. That's the time where we show the picture, and that's 1 second. Now, if you put these spikes in here, they are actually in here, you don't see anything. You can use any statistical criteria you want. We did. There's nothing significant here. If you see this neuron here, look at this. It fires very strongly to the Taj Mahal, but it's extremely sparse. This neuron is nearly silent at the moment we show the Taj Mahal fires. Now, what's the problem? The problem is mathematical problem, so that's a technical, say, session. The mathematical problem is that this neuron fired 200 spikes, 204 spikes, in about half an hour of a recording, whereas the multi-unit has 6,300 spikes, or this one fired 1,400 spikes. So a very tiny fraction of the spikes that you see here, they correspond to this neuron. Very, very tiny fraction. So it's very easy to miss this neuron, because it's like if I put a microphone here, and you are all talking and talking and talking, and there's one guy who has the truth of the universe, and he will say 42, and that's it. And he was, I think that was from the Hitchcock. So that's the answer to all the questions. And maybe later he will repeat 42. But since we are all talking from my microphone, I will never be able to distinguish that there's one guy that spoke only once and said 42. I will mix it up with the conversation from the others. But if I'm able to separate this guy, say, wow, look at this guy. He was silent the whole time, and then he, boom, gave the right answer. Now, why I'm stressing this too much? Because most, nearly, not all, but the vast majority of neurons, I will show you in the afternoon, they are like this one. So if you don't do proper spike sorting, you just don't see them. And I think that's one of the main reasons why nobody saw them before, because if you don't separate them, you will just merge them with neurons that are firing way too much. These neurons are nearly silent, and they only fire when you show the right picture, and in response only to this picture and maybe to something else. So that's why they're so hard to identify. But if we can do a good job with that, I mean, then suddenly we start seeing neurons that they were new, nobody saw before. And I think that's interesting side story of the spike sorting. It's boring because it's wavelets, superparametric clustering, the median, and so on, but if you get it right, and if you tune your algorithm right, it's all tuned. I mean, it's automatic now. But it took us some time to tune it. If you get it right, then you start seeing stuff, I mean, that is actually new. It's amazing. And I think, I mean, well, we have a lot of stuff, but if you are interested, if you do recordings, and if you are interested to check this algorithm, I will be here the whole week. I mean, we can see data and so on. There's a link from here, from our webpage. Again, there's some tutorials, introduction, simulated data, some examples from our recording. So it's very easy to play with it. And, oh no, I'm not doing this. OK, and I stop here. And so we have to say something. Sorry?
Perfect timing.
Yeah. Thank you. So you have a question?
Yeah, just one small point,
yeah.
Rodrigo. So the test that we always apply is to see whether one of our supposed single neurons ever fires within about 2 or 3 milliseconds.
Yes.
So, and if it does, then it's not a single neuron.
Yes.
Because of the refractory period.
Yes.
So that's a useful additional test.
Yes.
To put in, which I'm sure you do, but it may just be worth people knowing that that's really essential.
Yes. So what Edmond is saying is very important. If you plot the ISI distribution, so the time in between spikes, right? So if I get this cluster and I plot the ISI distribution, you see that there's a little gap here. And the number that the code gives you here is how many spikes you have firing within less than 3 milliseconds. Because if you have too many spikes firing within less than 3 milliseconds, this is a sign that you did a mistake. Because we know that a neuron, due to exactly what Edmund said, has a refractory period, cannot fire quicker than 300 milliseconds, right? Yes?
Sorry, can't fire quicker than 3 milliseconds.
300, no, 333, sorry. I just added the 100. 300 milliseconds is the latency of the response, but that comes later. I mix it up.
I have a question. When we are recording from one set over several sessions or several videos,
Yes.
how can I be sure that what I'm seeing in each day is the same thing which fired?
You want the quick answer? Never. No, the only way to do that is, which is what we're trying to do now and what some people are doing, is you have to record continuously. So basically, you have to track the neuron on a continuous basis, and if you see a radical change on the spike shape, you know that there's something went wrong. There's something wrong. Now, if you see smooth changes, so the spike shape, maybe if you take the first 5 minutes and you compare it with 4 hours later, the spike shape, they look very different. You say, well, it's a different neuron. But if you track the recording, and if you see that the change has been smooth, then it's fine. It's maybe like when you put an electrode inside the brain retracts, right? Moves slightly. You have to wait. You shouldn't start recording immediately. So how long should you wait is about what takes you to get a coffee. That's what every neurophysiologist will do. They go for a coffee, they finish the coffee, and then they go and start recording. But if the brain still moves, you will see changes in the spike shape. As long as they are smooth and you can track them, you can more or less safely say it's the same neuron. But in principle, I mean, you never have ground truth. It's very hard. Now, if there's no more questions, I think we have to arrange for the ones who are going skiing.